Hello students, we'll start with session 8 of paper Enzymology and Cellular Metabolism, and this video lecture I'll try to cover the clinical applications of enzymes. So enzymes are used in various fields, in industries, in medical field, in environmental biotechnology, in food biotechnology, and in dairy industry, in various fields. Okay. The therapeutic uses of enzymes we'll study one by one. So in this I have tried to cover almost 9 enzymes. Okay. So first one is asparaginase. So this enzyme, the therapeutic use of this enzyme is it is used to treat acute lymphotic leukemia. The cells need asparagine for its growth. Okay, but in the acute lymphotic leukemia, they are made to deprived. Okay, streptokinase, urokinase, again this plasminogen and streptokinase, they are used to break the clots or fibrin. Hyaluronidase, it is used to break hyaluronic acid and enhances the local anesthesia. And pancreatic lipase and trypsin, they are used to treat digestive disorders and pancreatic insufficiency by oral administration. And peptide is used, proteolytic enzyme used in the treatment of digestive disorders and also in the treatment of inflammation as an anti-inflammatory agent. Okay, and alpha antitrypsin, it is used in emphysema. Okay, we'll study one by one. The first one, asparaginase, is an enzyme that breaks down. See here the mechanism of action. Asparaginase, it is used to break asparagine into aspartic acid. Unlike normal cells, acute lymphotic leukemia cells are unable to make their own asparagine. Okay, but in a cancer cell, they cannot make asparagine. So asparaginase stops the conversion of asparagine to aspartic acid in the cancer cells that stops it from dividing and growing. This is the action. Asparaginase hydrolyzes the serum asparagine to non-functional aspartic acid and ammonia and depriving tumor cells of required amino acid. Thus tumor cells proliferation is blocked by interruption of asparagine dependent protein synthase. So asparaginase is used to treat acute lymphotic leukemia. It works by starving the tumor cells of needed nutrients and slowing the tumor cell growth. Oh, these are the normal cells and the cancer cells. When they are treated with asparaginase, it specifically kills the cancer cells by depriving them from aspartic acid. Okay, they are inhibited by their growth or division. This is the elaborative view where asparaginase is used to make asparagine, which are used to cleave the asparagine into aspartic acid and ammonia, and that is not happened in case of the cancerous cells where the cells are deprived of this asparagine enzyme, asparagine amino acid. Okay, and they are needed for protein synthesis. Here the protein synthesis is blocked. Okay, this is how it is used to treat the leukemia cells. Second one, streptokinase. The mechanism of action of streptokinase, it is used to cleave the plasmin. The plasminogen is an inactive form of plasmin. Okay, activator complex along with streptokinase and plasminogen, it is used to cleave the fibrin to soluble degradation products. It is a streptokinase. It is purified from the bacteria Streptococci. It shows proteolytic activity on human plasminogen into plasmin. Okay, streptokinase is a bacterial derived enzyme of serine protease group. It is produced by fermentation using streptococcal culture and is isolated from the culture filtrate. It is a thrombolytic medication enzyme. As a medication, it is used to break down the clots in some cases of myocardial infarction, that is heart attack, and pulmonary embolism and arterial thromboembolism. This is how it is therapeutically used. Urokinase, a human enzyme synthesized by a kidney and obtained from either from urine or cultures of human embryonic kidney cells. It is a direct plasminogen activator. It is given intravenously. Urokinase is a fibronectic enzyme produced by recombinant DNA technology also. from E. coli cells. It is urokinase is medicinally it is also purified directly from human urine. It binds to the range of adsorbents such as silica gel and can be used to initially concentrate and purify the product. So it is also purified and precipitated with sodium chloride, ethanol by chromatography. The urokinase is available commercially in this way, used clinically as a thrombolytic agent in the treatment of severe and massive deep venous thrombosis. The peripheral arterial occlusive disease and pulmonary embolism, acute myocardial infarction, and in the occluded dialysis canals are catheter clearance. This is how it is available commercially in the market for the thrombolytic agent. See here, it is a artery where the clot, you can see the fibrin clot here. Okay, the enzymes urokinase along with this tissue plasminogen activator, which acts on this plasmin, okay, and cleaves the fibrin to form a fibrin degradation products. Same thing I have shown in the flow chart. The next one is that is Serato Peptidase, is a proteolytic enzyme isolated from non-pathogenic enterobacteria. Serato is the most widely prescribed anti-inflammatory enzyme in the developed countries and also in India. It is used to eliminate inflammatory edema and swelling, and it accelerates liquefaction of pus which is formed around the wounds. Okay, it enhances the action of antibodies. It is also used to for fast healing agent. It is proving to be a superior alternative to the non-steroidal anti-inflammatory drugs traditionally used to treat rheumatoid arthritis and osteoarthritis. Okay, it is widely used in trauma surgery, plastic surgery, respiratory medicine, and obstetric and in gynecology. This is how it is available. Okay, it is, as I told you, it is used as anti-inflammatory agent, analogist, fibrolytic agent, and antibacterial agent. Hyaluronidase, it is also one of the enzyme. It is used to break down the hyaluronic acid. Has long been used to increase the absorption of drugs into tissue and to reduce the tissue damage in case of extravasation of drug. It is epidermis hyaluronic fiber, where when you that is intake this hyaluronidase enzyme, it will degrade this dermis. Okay, and you can see the hyaluronate fragments which is required for spreading or diffusing the substance. It increases the permeability of connective tissue through the hydrolysis of hyaluronic acid which is present in the epidermis and dermis. Hyaluronidase enzyme, it hydrolyzes the hyaluronic acid by splitting the glucose amide bond between carbon 1 of the glucosamine moiety of C4 of glucuronic acid, and this is how it is available in the market commercially. Next, lipase, it is used in the digestive disorders. It hydrolyzes the dietary fat molecules in the human digestive system. It is one of the main digestive enzymes converting triglycerides, oils and fats into monoglycerides and free fatty acids. You know this because in the last semester I have studied about oils and fats. So in the intestine, the dietary fat is broken down by pancreatic lipase so that they are absorbed by the intestine. Thereby, it qualifies as the most vital enzyme for digestion of dietary triacylglycerides. Thus, inhibiting the pancreatic lipase will reduce the amount of fat absorbed by the intestine. So commercially also it is used in the food industry and in the dairy industry also for flavor enhancer. They can also improve the characteristic of flavor by cheese acting on the milk fats to produce the free fatty acid after hydrolysis. The trypsin is also used in the treatment of digestive disorders where for the digestion of proteins. Trypsinogen it is an inactive form of trypsin. When it is activated, it is converted into that is active enzyme for digestion of proteins, which converts the long polypeptide chains and converts specifically between the peptide bond between arginine and lysine. Trypsin catalyzes the hydrolysis of peptide bonds, breaking down the proteins into smaller peptides. The peptide products are then further hydrolyzed into amino acids via other proteases, rendering them available for absorption into bloodstream. It is also given to the people who lack enzymes needed for digestion. It is also given in combination with bromelain and rutin for the treatment of osteoarthritis. Some people, they apply trypsin directly on the wounds and ulcers to remove the dead tissue and improving the healing process. The next one is peptide. You heard of, these are available in the market as tablets. It's isolated from Carica papaya. It's again a proteolytic enzyme helped to break down the proteins, in the process of which it makes ideal for meat tenderizers. That means the muscles, they are make it smooth by using this proteolytic enzyme. It is also called a ideal enzyme for meat tenderization. Peptide is a popular folk remedy to reduce pain, inflammation and swelling. It is also been used to improve the digestion and to treat infections, diarrhea and allergies, and is also being studied for potential use in the cancer and other disease also. This is how it is available in the market. The same thing it is have repeated here. The peptide, it is extensively used as a meat tenderizing agent and in meat packing. It is a proteolytic enzyme which, similar to the pepsin, it is combined with other enzymes such as amylase to produce digestive aids. It is also used to treat the contact lenses of prolonged wearing time and keratonic patients with papillary conjunctivitis. The last one is SGOT and SGPT. These are the two enzymes which are present in the serum. Okay, that is glutamate pyruvate transferase and alanine. It is also called alanine transaminase. Is widely distributed in the cells throughout the body. This GPT, that is glutamate pyruvate transferase. It is found predominantly in the cytoplasm. It is widely considered to be specifically for liver. In addition, it is also active in the heart, skeletal muscle, pancreas and the kidney. If there is a damage in these vital organs, the level of this enzyme, it shoots up. Okay, and this ALT, alanine transaminase, activity in the liver is about 3000 times higher than its activity in the serum. Only if the cells are damaged, it will be excreted into the bloodstream. So in the plasma, ALT has no biological function because the necessary substrate and co-substrates are lacking there. It is a reaction what they are, how they are acting. That is, aspartate reacts with alpha ketoglutarate to form oxaloacetate and glutamate. It is catalyzed by the enzyme aspartate transaminase. So it is a cofactor. Vitamin B6 acts as a cofactor. Another enzyme, alanine transaminase, here it is alanine and alpha ketoglutarate. It forms a pyruvate and glutamate. Okay, these are the two enzymes which they release into the bloodstream only if there is a injury in the. So here it is a liver cells. When the mild injury, it changes the cell permeability and increase in the level of these enzymes. The clinical significance. It is raised in the acute liver damage. It is also present in the red blood cells and the cardiac muscle, skeletal muscle, and may be eliminated due to the damage to those sources as well. Aspartate transaminase was defined as a biochemical marker for the diagnosis of acute myocardial infarction. However, the use of aspartate transaminase for such a diagnosis now reductant has been superseded by cardiac troponins. Okay, and aspartate transaminase is commonly measured clinically as a part of diagnostic liver function test to determine the liver health. So this is the normal level of the serum, that is alanine transaminase. It is present less than 45 units per. Okay, thank you for watching. Keep watching.